Hey, we're rolling. So it's my job to convince you that it's a microbial world. Title of the talk. So we're surrounded by microbes and they play a really, really important role. And we actually know very little about what role they do. And the second topic of my talk is all about trying to disprove what Alicia said yesterday, and viruses. It's all about the bacteria. So it's going to be a long story. I'm starting in 1680, so you could be here a while. So 1680, Anton van Leeuwenhoek was the first guy to see a bacterium. He was actually a lens maker, so he made these lenses, and he was the first guy to invent the modern microscope. So he's called the father of microbiology, basically, because he actually looked down these lenses and saw little individual cells. That was the first time anybody actually saw a bacterium, and that was in 1680. If we go forward to 1976, NASA sent a probe, the Viking lander, up to Mars, and that was there to actually look for life on Mars. So in 1976, they were looking for life on Mars outside our planet. But we didn't actually know about 99% of life on our planet as it stood. So it wasn't until, well, a bit later that we actually understood anything about microbial life. So 76, we're going to Mars, going to other planets to look at life on other planets, and we don't know anything about life on our own planet. And that's because everybody had been looking at life-threatening diseases, so things that kill us. And that's understandable. So for the last 2,000 years, we've been trying to understand what actually kills us. From 1850, when they discovered cholera, like the cause for cholera, that was actually a major breakthrough, a major step. And there's been major advances through with the finding of antibiotics, for example. But it wasn't until 1977 they actually termed the coin microbial ocean, because nobody actually thought anything lived in the ocean. They thought it was completely clean. It was an oligotrophic environment, so nothing could live in there. But they're actually hugely amazed when they looked at took a mill of seawater, and there was 1 million bacterial cells in 1 mill of seawater. So you go out here and take 1 mill of seawater, you'll see 1 million bacterial cells just in that 1 mill. And nobody had any clue until 1977. So that's how recent it is, actually really recent. Like it's 35 years, basically 36 years since they discovered the microbial ocean, and microbial oceanography was actually born. So, like I said, 1 million cells in every mill of seawater, and they remained undiscovered because they're actually extremely small. Nobody actually saw them, nobody actually had the technology to look at them, and they didn't think they were there at all. They thought it was nothing.
If you took out those million cells from 1 mill of seawater, how much seawater would you have?
I don't know, that's a good question, I don't know. I mean, you could actually probably do the calculations, but I'd be guessing.
Because it'd be made might be extracted from the ocean.
Yep. Yep, absolutely.
By mass, bacteria have the biggest components.
Yeah, and I got the figures in here, especially in the oceans. So this is a tree of life, and this is actually a basic tree of life. So if you look at it, it's broken up into 3 kingdoms: archaea, bacteria, and eukarya. Us, human life, multicellular organisms, cows, horses, sheep, anything you can think of, monkeys. They all fit on those twigs of the tree of life. Everything else is single cellular. Everything else is microbial. So in terms of the biodiversity on our planet, it's all microbial. The biodiversity that we see is actually just very minor in terms of the whole scheme of things on our planet. So it's all single cellular, mainly, all microbial, things that you can't see. So most people go in the marine environment and look for dolphins, turtles, sharks, etc. They're the megafauna, the charismatic megafauna, but they actually make up very little of the actual biodiversity in our oceans. So if you look at that pristine coral reef, that's what we think a coral reef should look like. There's a whole heap of biodiversity on there, but you actually can't see most of the biodiversity. So what you see there is actually not what you see in terms of whole biodiversity, because it's all microbial. So if you actually scale down in the coral, you see the coral fragments there, lots of zooxanthellae that we've been talking about this week, and then you get into the microbial fractions of the bacteria, the viruses, the protozoa, all the single cellular organisms. That's where the majority of the biodiversity is. So if you look at the estimates, there's about 2 times 10 to the 6 species of bacteria in the oceans alone. So that's just a general estimate. Nobody really knows how many bacteria are actually out there. There's about 5,000 bacterial species have been cultured out of that number. So not very many. It's an enormous reservoir of unexplored biodiversity. that we're just starting to tap into, that we're just starting to understand, and equates to unique activities. I mean, there's these whole range of functions that they do that would be really important to actually look at and see, because they do amazing jobs that we wouldn't be on this planet without them being there. So the global oceanic densities, like they're the numbers, like huge numbers in terms of the number of bacteria, archaea and viruses. So if you put all the viruses on end to end, it's stretched to the moon and back in terms of that's how many viruses are actually in our planet, and that would make up the distance that you could travel from the moon and back if you just put them end to end to the moon and back. There's that many viruses and we have no idea what they do surrounding us. So we have so little idea about them. So the microbes account for about 50% of living protoplasm on our planet. So in the oceans, there are about 98% of our biodiversity is actually in the microbial realm. So huge numbers. They cycle 10 times more nutrients than plants. So we think the plants are the lungs of the ocean. We often talk about the rainforest being the lungs of the ocean. It's not true. It's the oceans. And it's the microbial organisms in the oceans that actually are cycling all these toxic gases, cycling all the nutrients, cycling carbon, nitrogen, phosphorus around the planet. The microbes do it. And it's a very good reason. So if you look at that timeline, I mean, geological time frames in billions of years, I mean, microorganisms appeared about 3.5 billion years ago. Humans are about 3.5 million. So we actually aren't even on that scale. We're right at the end of that scale. It actually doesn't even register in terms of how long humans have been on the planet compared to microorganisms. And if you go back in terms of arthropods and dinosaurs, you can see where they appear. So these microbes have had billions and billions of years to adapt and change and develop in their biodiversity. So that's why they're so abundant and they're so amazing in what they do and actual things they can do. So the ocean, our atmosphere. So the cyanobacteria we looked at last night is related to the organisms that actually gave us the oxygen in the atmosphere, that created a planet that we could actually inhabit and that multicellular organisms could actually grow out of. So, because they produce 99% of the oxygen in the atmosphere, all derived from cyanobacteria. So blue-green algae, what we saw under the microscope, they were the guys that actually put oxygen into our atmosphere through their metabolisms, because they arose at a time when there was a highly anoxic Earth environment, and through their metabolism and the way they evolved, they're actually able to produce oxygen and made it a livable planet. So, and they gave rise to the O2, and it's well documented in the glaciation, that process. So you can actually see it in the whole ice cores that have been dug through Antarctica. So geomicrobiology, there's a huge field in geomicrobiology at the moment. And the reason is you're going to dig almost to the centre core of the Earth and you'll find microorganisms. You'll find bacteria living there that are alive. So you can drill 3-5 kilometres, no matter how far people have drilled, they pulled out cores and they're able to grow bacteria and observe bacteria. So there's guys down there that live really slowly and do quite amazing things. All our mining is basically derived from microorganisms. So the laying down of certain forest degradation processes that give us our aluminium, our iron, etc. It's all through microbial processes over geological timescales. So our mining boom at the moment, we can actually thank microbes for as well. So waste treatment, all our waste treatment is basically dependent on microbes, on bacterial processes. So we'd be swimming in our own shit, unless it was for the microbes, basically. Those toilets that we're using at the research station, so they're biocomposters, they depend on the microorganisms there to break down the whole process in our waste products. And these guys love it. I mean, they will break down rich nutrient environments and process these waste that we consider waste into something that's reusable, and actually make it a pleasant thing to actually elute. You can get zero discharge from sewage treatments if you actually know the microbial processes and implement them. And that's why there's so much research going into waste processes. There's a huge field of microbiology, is just to understand the degradation of waste and in sewage plants, and actually get zero discharge of nutrients out of waste plants. All microbial processes. So radioactive cleanup as well. So the nuclear reactor in Japan, for example, I mean, the only way they can clean that up from the cesium degradation, from the cesium contamination, from the radioactivity, will be through microorganisms. So this is a guy, Dinococcus radiodurans, can tolerate huge amounts of radioactivity. So it's been engineered to actually take out and biodegrade radioactivity contamination. So what may happen in the Japanese disaster what they'll be looking for is actually taking some engineered microorganisms and trying to remediate that, because that's the only way they can do it over a time frame that's human lifespan. So getting
So this one is
naturally high radioactivity,
You
yep.
can improve it.
But you can actually engineer it to actually have higher radioactivity and tolerance, but also more rapid degradation.
So it breaks
cesium.
down the radioactive element of the sun as
Yeah.
well.
Yeah, so you get byproducts that are radioactive, but then you have the whole process. So, and cesium's a problem, like in the radioactivity in Japan. And amazing things, like you just have to go into the animal world, and termites are amazing animals. So a large amount of our biomass on the planet is plants. It's cellulose, which cannot be broken down. So ruminant animals have these unique systems within four guts, for example, that have microbial processes going on to break down cellulose, because it's so resistant to any sort of breakdown. Termites have this hind gut that have these amazing microbial communities that process cellulose at rapid rates. So by studying the termites, they're actually trying to engineer the ruminant guts, so for cows, sheep, etc., so that they break down the cellulose that they're eating, the grass, the hay, etc., faster, and it gives greater biomass onto the animal itself. So studying termites can improve cattle production. And so they basically inject microorganisms into the cattle to actually get faster cellulose degradation and better conversion of cellulose into meat, for example, that then we eat. So, but also a huge amount of study looking at, well, methane. Basically, cows produce a lot of methane. These guys have amazing microorganisms that can metabolize that methane and reduce the actual methane emissions from cows, etc., which are a major emission in global greenhouse product as well. But then also new energy processes also. So, and the human gut. So sitting there, you're actually more a bacterium than you are human. So you've got 10 times more bacterial cells in or on you than you have human cells. So you're actually a bacterium, really. So about 5% of your body mass is microbial. So if you took out those microbes, it'd be 5% of your body mass. So they're doing really important things in your body. So there's been some studies where, for example, obesity. You can take a fat mice and a skinny mice, you can swap around their gut microflora, so the bacteria within their gut. The skinny mice will get fat, the fat mice will get skinny. So a lot of metabolism depends on the actual organisms within your gut. So there's a lot of studies looking at gut microbiota actually as a cure for obesity problems as well, because they're the ones that metabolize a lot of the saturated fats. And if you can actually manipulate and change your gut microbiota, you can actually metabolize things faster so it doesn't get fatty deposits on your body. So there's a lot of work trying to understand those processes, for example. And it's much more complex than that. There's a whole heap of lifestyle choices as well. But your gut microflora has a very important role in whether you're a skinny or a fat person. So in your metabolism rates, basically. Really interesting stuff. The Japanese, for example, they've got this community of microflora in their guts that allow them to eat seaweed and metabolize seaweed. Most people in the Western world can't eat seaweed and metabolize it. The Japanese can. They've co-evolved with their microorganisms that they've eaten seaweed for so long over their lifetime and through generation after generation that they have a different microflora that actually processes different cellular foods and actually converts it differently. So it's really important. There's a whole huge study looking at the human microbiome, trying to understand the whole microbiome of people and what role it plays. Because it also plays important roles in certain diseases like Crohn's disease, for example. It's your microbiota that's causing that. And we heard last night, for example, viruses, they play an important role in cancer. So some of the cancers are caused by viral infections. So it's not just a simple mutation of your DNA, it's actually a viral infection in some cancers. So really interesting stuff.
So yeah, cool. That's really, it's actually good for you.
It's good for you, yeah. So having a balanced microbiota is really important. Antibiotics for kids is really bad. So, because kids grow up with a microbial community and they have to get exposed to that microbial community. It's generally passed on from the mother to the child through breast milk, and it's an inoculation process. So as soon as kids go into antibiotics, especially very young kids, you upset that balance between your microbiota and your human body, and that can have some really dire consequences later in life. And there's correlation between antibiotic treatment in kids and all sorts of allergies and health problems later in life. for kids that are exposed to long-term antibiotic treatments. So, because they've just upset their whole microbiota. So really important roles in the whole human health system. Nitrogen, for example. This just gives an example of what important role they play in our terrestrial and marine environments. So nitrogen is 78% of the gas in the atmosphere, okay? It's not very biologically available, so nothing can really use nitrogen except for a few plants here and there. The microorganisms, the bacteria, are the ones that actually convert nitrogen into something that's biologically usable, especially in the marine environment. So it's the cyanobacteria, it's the other diazotrophs, they're called, sorry. So single cell microorganisms that fix nitrogen. And what I mean by that is they take nitrogen gas and convert it into ammonia. And then that's passed through the actual trophic system. So it gives nutrients, because everything needs nitrogen. If you don't have nitrogen, cells can't live, cells can't do what they do. It's the bacteria that convert the nitrogen into something that's usable within the marine system especially, and in many cases within the terrestrial system. And they cycle it through nitrate, nitrite. It's all a microbial process. Without those microorganisms, the world wouldn't exist, because nitrogen would just get used in one form and nothing could get remineralised, and no living system could actually use it without the microorganisms. So we're only on this planet because the microorganisms let us be here, basically. They're the ones that are doing these key drivers. So another example is through some sequencing technology in the ocean, they found this gene, photoredoxin. So basically they thought light from the sun was photosynthesized by algae, and it converted into the algae biomass, and that fed the food chain. So it's probably about 15 years ago now, they found this protein within bacteria. So within single cell bacteria in the ocean, it's called the SAR11 clade, they make up about 50% of the biological biomass of the oceans. This protein takes light and converts it directly into energy. So it actually changed the whole global climate budgets, the whole cycling budgets within the Earth, because they found this through a sequencing approach, and all of a sudden there's 50% of the biomass that they didn't know about, 50% of the organisms in the ocean are converting light directly into energy and passing that through to different trophic levels in the marine system. So it was only discovered 15 years ago, and it's the major driver and major fixation of light into energy within the Earth, basically, through the SAR11 group of bacteria. So that's kind of how recent the discoveries have been made. So every year there's these huge discoveries in the microbial front, because we have so little understanding of what role they've been doing and who they are. And we actually now have technology and understanding to go out and look for them and see what's actually happening within the marine environment, but also the terrestrial environment. So all our understanding of global climate change processes, it's all done on physiochemical models. But it's actually the biological systems that have a huge effect on these processes. And we know so little about it that we don't know whether they're going to exacerbate or mitigate the effects of climate change, increase CO2, etc. So it needs to be factored into some of the models in terms of what's been understood. So this is a great quote. There's a guy in Newcastle in England, and this is a quote that he wrote. And it's a great one because it's so true. It says, "I make no apologies for putting microorganisms on a pedestal above all other living organisms. For if the last blue whale choked to death on the last panda, it would be disastrous, but not the end of the world. Like, we'd still go on. But if we accidentally poison the last two species of mono-oxidizers, that would be another matter. It could be happening now and we don't know about it." So it kind of says that these microorganisms are actually doing things. They're processing our waste that could never be done by anything else. And if they're not doing that, then the world would cease to exist as we know it. Our life would actually just peter out. We'd die in our own waste. So the microorganisms are doing exactly what we need them to do. And we're here on the Earth because they're letting us be here, basically. We co-evolve with them, and they're doing fundamental, important things for us. So, and the reason we didn't know a lot about them is we studied them in the lab. So we've always studied them in the lab. We've always tried to grow them. The problem is you actually can't grow a lot of the biodiversity out there. So about 99% of the organisms, 97% of the bacteria in the environment can't grow because we thought we'd grow them on rich media. They actually live in this oligotrophic environment. And so you can't actually grow like you do in medical systems where they try and grow the disease-causing organism. So you have to have different methods to try and actually get to them and try and understand what they're doing and who's out there. And there's been a whole range of new tools, and I won't go into them, but basically sequencing technologies. In the last 15 years, there's a whole revolution in the biological field through sequencing. So just as we sequence the human genome, our DNA blueprint, we now can sequence everything. We can go out there and sequence whatever we want, very cheaply, very affordably. And the reason for that and what's driving that is these companies want to sequence your DNA. They want to charge you $2,000 to sequence your DNA because then you get a blueprint of what genetic disorders you have. So it's very important. You can actually go and say, I'm predisposed to a certain disease, for example. I'm predisposed to bowel cancer. I'm predisposed to whatever, Crohn's disease, etc. So people can actually get a blueprint of what health problems they may have in their future, which has some interesting ethical implications about them in terms of managing your medical privacy, especially for insurance companies, because obviously insurance companies may not be able to insure you from health benefits if you're predisposed to certain diseases. It doesn't mean, if you got your genome, it doesn't mean you're going to get that disease, but you may be predisposed, you may have a high chance of getting it. So it gives you a chance to manage disease later in life. So for $2,000, you can sequence your genome and you'll have a blueprint of what it is and get a printout of what diseases you may be predisposed to. And then get one for your partner, and you can look at what your kids may have if you have kids. That's here now. That's the reality today, because that's what the companies are doing. So you can walk now in and take a swab from your mouth, send it away and get your genome sequenced, and you'll get it back within 2 weeks. And you'll have a blueprint of your whole genome. No. No. So there's some interesting ethical questions behind it, and that's what they're struggling with. The technology is moving so fast that they're trying to catch up from that ethical point of view. But companies like Illumina and Solid are the two major ones at the moment, and that's what they're developing the technology for. Is the Craig Venter.
Yeah. I know he's now sampling 100 kilometres,
Yeah, so he did the Voyager 2 cruise across the world. So every 100 kilometres, basically sampled the ocean, took 100 litres of water, filtered it, and then sequenced everything in that seawater sample. So basically sequenced every single organism, bacteria, viruses, cyanobacteria, etc., just to get an understanding of what's actually out there. And through that process, they came up with 7,000 new species that they never knew of, 10,000 new protein families that they never knew of. So there's this whole range of new discoveries, and that was probably 8 years ago now, I guess. Venter went around the world. Yeah, basically. Still done, yeah. Right. Okay, so that's, again, Sargasso Sea sequencing and the Venter cruises. So DNA sequencing, we're in a revolution at the moment. So, and it's quite impressive. The speed of the technology is changing so rapidly, it's really hard to keep up. And people are struggling to keep up with the data. So there's so much data being able to be generated. How do you store that data? How do you analyse that data? That's a real critical question at the moment for a lot of the biological disciplines. And I will put viruses in there. So I said 1 million cells of bacteria in every mil of seawater. There's actually 10 times that many viruses or phages. There's about 10 million viruses in every mil of seawater. So you consider how many mils of seawater are actually out in the oceans. That's a lot of viruses. And they're playing a really fundamental role. And as Alicia says, we don't know much about what they're doing, all those viruses. So we know they're important, but we have very little idea about how important they are and in the exact roles that they're playing. So metagenomics. So that's what the girls are actually doing here. So Alicia, Karen and Kathy, they were here because they were sampling corals, and they're going to do this metagenomic approach. And that idea is actually just sequence everything. Sequence the whole microbiota, the microbiome of the coral. Just as there's human microbiome projects, sequencing every single bacterial cell in the human from 10,000 individuals, there's also activities to sequence the coral holobiont microbiome. And all it does is basically you just isolate DNA, you chop it up, randomise it, and then you just sequence it. And the technology's there where you just get massive parallel sequencing, so you get millions and millions of sequencing reads. So Yui just got a data set for his transcriptome, like he said, 80 gigabytes. So that's an enormous amount of data. Like trying to sort through that amount of data and figure out what it actually means is really quite challenging. So if you want a job and you want to earn well, become a bioinformatics person. That's a computer science person that looks at DNA, basically trying to put DNA back together. That's in essence what the actual process is. So corals, just to finish up on corals and the microbiota of corals. So like Yui said, corals are a holobiont. We're a holobiont. Every organism in this planet is a holobiont, which means it's not only the organism, it's actually the bacteria, the protozoa, every other single organism associated with it. It's all co-evolved. So the microorganisms have been there for billions of years before any multicellular organisms actually appeared on Earth, and they've all co-evolved. So there's this, and it seems pretty obvious when you think about it, but it's only in the last 10, 20 years that they actually realised that actually we're a holobiont. We're not just a single organism, we're multiple of organisms. So it's really a radical change in how we look at all organisms on the planet. So you have to understand what role they're doing. And I've been saying humans, huge. Your 5% of your body mass is bacteria, you're 10 times more bacteria than human cells. In corals, it's even twice that. So it's 100 million cells per centimetre squared. So there's huge amounts of bacteria on corals. So even 10 times what's on the human body. So they must be doing something really fundamentally important for that animal. And we have no idea what the bacteria are really doing. So we have very little idea. We're just starting to look at that at the moment. So what are their functional roles? I mean, what are they doing, basically? And this is one question. I was talking about nitrogen before. So if you look at corals, they're the host, they're the Cymbidium. So basically the Cymbidium provide energy to the coral. So they photosynthesize light, convert it into something that the actual host can use. The host provides a really nice home for the Cymbidium. So it's a symbiosis, it's a classic symbiosis where those two work together. But you also got all these other microbiota in terms of what are they doing as well. They're doing a really fundamental role for corals. Probably antibiotics, probably yeast exclusion, and they also function in the health state of the animal. But there's also interactions between the Cymbidium and the microbiota as well. So it's a complex three-way partnership. And if we look at some of the sequences, this is some work from Kim Lima, who's a PhD student, been working with us, and she looked at diazotrophs, basically guys that fix nitrogen. So I was saying nitrogen is really abundant in the atmosphere, but it's not biologically usable. You've got to get it into something that's biologically usable for organisms in the marine system to use, because the marine system is really nitrogen limited. So the only way it gets nitrogen is through fixation through bacteria, called these organisms diazotrophs. When we sequenced bacteria associated with corals, about 67% of sequences that we retrieved were related to this group of rhizobia. And that's really interesting because if anybody remembers their basic biology courses through school, plants have nodules that are actually full of bacteria that fix nitrogen, the rhizobia. So these plants basically have this whole group, nodule, bacteria, take atmospheric nitrogen, fix it, passage it to the plant. What we found in corals was exactly the same. There's all these bacteria in corals that actually are related to the bacteria in plants. They're part of the rhizobia group. So they're taking nitrogen and they're fixing it and passing it into the coral itself, which is a really interesting functional role. So for 50, 60 years, I've been studying plants and looking at the rhizobia, and we now know the same thing's happening in corals. And that's interesting because the corals are sort of like a plant as well. They've got the algae there, and they need that nitrogen, and that can only come from the bacteria. So that's one of the functional roles we've been looking at in terms of fixation of nitrogen. So that's about it. Quick tour of why microbes are important. Any questions? So from the work we do, I mean, Yui's doing obviously disease, but there's a real big interest in actually the beneficial role of bacteria as well. So 99% of bacteria in your body is doing a beneficial role. The ones that cause disease are so minor, they're a very minor constituent. And the majority of the time, those disease-causing organisms, you're not their target. So basically, what their target is some other ecological role. And Vibrio cholera is a classic example. So we all know about cholera. So cholera comes in contaminated water, people get sick, diuretic cholera causes all sorts of massive problems. It's actually a marine bacterium, has a very different ecological niche. So it associates with a lot of organisms that have chitin in the marine system, and that's their ecological niche. They're there to actually just exist with these other organisms in the marine environment. When you get, mainly copepods, so when you get elevated seawater temperatures, you get outbreaks of cholera in Bangladesh, India, etc. The reason for that is you're getting massive copepod blooms, which are increasing the Vibrio cholerae because there's a host there, and that leads to contamination in freshwater systems in Bangladesh. So they can correlate seawater temperature, copepod blooms, Vibrio cholera infections. So it's just serendipitous that the cholera infects humans because that's not their target host. It's actually the target is the copepods.
So if the coral reefs continue to die the way they are, then the nitrogen in the atmosphere is going to go up, which means humans might survive like this at the end of the day.
Probably not. Then you'll get a feedback of bacteria actually being able to metabolize more. So at the end of the day, the microbes will survive. They don't care what happens on this planet. If we disappear, they'd be quite happy and just keep going on their own little route. But there'd be feedback loops there, without a doubt.